We don't know yet how much of that permafrost carbon will go up to the atmosphere as carbon dioxide or be exported to the Arctic Ocean and basically stay there for a longer amount of time.
It starts off with you take a sample from a thermal karst impacted water or a surface water. We actually bring the sample back to lab, we measure all the different nutrients, and we characterize the carbon that is in the solution.
Set up these large experiments on the tundra where we'd expose it to the natural sunlight over the course of a day and basically measure the amount of CO2 produced.
Right now we're just labeling sample bottles, and each bottle will be filled with filtered water. They each get a different color of tape because each bottle in these lines will be analyzed for a different component or analyte in the water. Most of them just end up getting shipped home to our labs in Michigan, where we'll run them on machines down there because we don't have the equipment to do it up here.
And we collect, I don't know, 1,500 samples over the course of a season.
In our experiments, what we do is we take water, like from the Kupark River or somewhere, we filter it, and we take that and put it out in the sun and just let it be photo exposed, or let it soak up sunshine, essentially.
Initially, when it comes out of the soil, it's really good at being turned into carbon dioxide by sunlight. And we think that's just because this is carbon that's never seen sunlight before.
And we also then take the same water and keep it in the same exact treatment, but then we wrap it in aluminum foil and leave it out there right next to it, so it's kept dark, completely dark.
There's all these what we call labile molecules, or when it's labile, that means that the reaction is fast. You know, this is stuff that can be easily converted into carbon dioxide.
We take that water in and we essentially feed it to bacteria and look at what they do with it and how active they are given these two types of carbon, this light carbon and this carbon that was kept dark. So what I've measured was bacterial production, which is a snapshot of just how active those microbes are.
The difference isn't in the process of the bacterial production, the difference is in the carbon that is used to feed the bacteria.
Right. Mm-hmm.
So the sunlight changes the carbon and makes it more readily used by the bacteria, whereas compared to the carbon that was not exposed to sunlight. So that's the difference between light and
Right.
dark.
You can think of it like how well the food is baked for these microbes. So if you take a frozen pizza out of the freezer and just give it to these microbes, they don't like it. But if you take it and you put it out in the sun and bake it for a little while, they'll eat it a lot faster and turn it into CO2 a lot faster.
Mm-hmm.
What we don't know is if you take carbon coming out of the soil, how much of that is the easily converted carbon dioxide. 50% of the organic carbon in the soil? Is it 10%? And that's something that we're trying to get at with our experiments.